speech only, we realized not only are we posted with the reasons of advancement of building biofactory with so bright ideas, but we came into realization that some living reservoir, especially those very near to us, worth digging up scientifically, and that might be commercially or economically gain for us. Thank you so much. And next is the photo session, right? Photo sessions. Participants who are attending, please unmask yourself if you are wearing one, because that's not, yeah. This is things to remember. Who are going to click the pictures, the committee? How many layers are we? 4, 5, or 1 layer? On my count, 3. On my count on 3. 1, 2, 3. All good? Thank you so much for all participants. We now move on to the discussion sessions, which will be led by moderators. For first sessions, our moderators is Dr. Sabar Pambudi, where he is acting as our chair for vaccine research group. So Dr. Sabar, floor is yours.
Thank you, Dr. Ika. Ladies and gentlemen, it will be for the next session today for invited speaker. So before I start, I would like to introduce our speakers. So this morning, one invited speakers from Shizuoka University will deliver the one presentation. His name is Dr. Takatsugu Miyazaki. He graduated from Tokyo University of Agriculture and Technology from for his bachelor, master, and PhD degree. After he graduated from TUAT, he got some position from Japan Society of Promotion Science, and he continued as assistant professor in Research Institute of Green Science and Technology, Shizuoka University, from 2016 until now. So he has a lot of publication, and today he will share about one topic. The title of the presentation is Structures and Function of Novel Glucosidates Discovered from Bacterial Genomes. So I would like to invite him to give the lecture. So Dr. Miyazaki, the time is yours.
Thank you for a kind introduction. Good morning, everyone. I am Takatsugu Miyazaki, an assistant professor of molecular and biological function research core, or RIGST, Shizuoka University. First, I would like to thank the committee for giving me the opportunity of my presentation. So, as introduced, I obtained a PhD at TUAT and joined RIGST at Shizuoka University 6 years ago. I am interested in carbohydrate active enzymes like glycosidases and glycosyltransferases and their 3D structure. So I have been studied protein glycosylation and glycoengineering with Professor Park, and microbial and insect glycoside hydrolases using enzymological techniques and protein X-ray crystallography. Recently, I started to use cryo-EM for protein structure analysis. I now belong to Molecular Biological Function Research Core at RIGUST. Our research core aims to contribute to the achievement of SDGs number 3 and 9 by technology development for the application of biological and molecular functions to improve health and extend healthy life expectancy. My team analyzed three-dimensional structure of proteins of interest. Well, today I would like to introduce my recent studies on two novel glycoside hydrolases. One is alpha-1,2 glucosidase, and another one is alpha-1,3 glucosidase. First of all topics, I will explain the research backgrounds and purpose. Carbohydrates are involved in various biological functions. Energy storage materials such as starch and glycogen, osmolytes, cell walls, and shell components like cellulose and chitin. And intercellular and intermolecular communications involve protein N and O glycosylation in mainly eukaryotes. They are also related to pathogenicity, diseases, and cancers. Moreover, some carbohydrates, especially oligosaccharides, act as prebiotics that induce the growth of beneficial intestinal microorganisms. Glycoside hydrolases, GHs, are enzymes that catalyze hydrolysis of glycosidic linkage in glycoside, oligosaccharides, and polysaccharides. They are involved in degradation, synthesis, and reconstruction of carbohydrates. For example, starch is a glucose polymer. It is hydrolyzed by alpha amylase and produce malto-oligosaccharides, also called dextrin. These oligosaccharides are further hydrolyzed by alpha-glucosidase to produce monosaccharide glucose in our bodies. Some GHs has transglycosylation activity and form oligosaccharides. So they can be applied to synthesis of various oligosaccharides. There have been reported more than 200 GH activities to date. GHs are classified into GH1 to GH173 families based on their amino acid sequence homology and summarized in the KZ database. Each family contains GHs with different activities and substrate specificities derived from various organisms. Some families contain viral GHs. For example, GH31 contains alpha-glucosidase, alpha-xylosidase, and alpha-galactosidase. Each GH family contains a tremendous number of putative enzymes encoded in the genomes of various organisms that had not been characterized yet. So we are aiming to discover GHs with novel activities from mainly microorganism genomes and to elucidate their structure-function relationships. Okay, next I would like to present the first study, structure and function of GH65 alpha-1,2 glucosidase. GH65 is a special family which contains not only GHs, but glycoside phosphorylases, GPs. These enzymes are active on alpha-glucosidic linkages. GHs cleaved glycosidic bond using a water molecule. In contrast, GPs cleaved glycosidic bond using inorganic phosphate and produce glucose 1-phosphate. GPs catalyze not only phosphorylases, but also reverse phosphorylases. So they can be applied to synthesize various oligosaccharides. Therefore, we became interested in GH65 family and searched for a new enzyme. This is the phylogenetic tree of GH65 enzymes. GH65 is divided into the bacterial GPs and eukaryotic GHs. All bacterial enzymes are GPs acting on mainly glucose disaccharides such as maltose, trehalose, cogibiose. And all eukaryotic enzymes are GHs, like trehalase. We found a protein gene which has low sequence identity with both bacterial GPs and eukaryotic GHs in the genome of soil bacterium Flavobacterium johnsoniae. So this was targeted. We cloned the gene from this bacteria and expressed in E. coli as a recombinant protein and purified, and then investigated its activity for glucose disaccharides, described here. We analyzed using thin layer chromatography, unacceptably, this enzyme degraded only alpha glucobiose, cogibiose, and produced only glucose, but not glucose 1-phosphate, unlike other non-bacterial GPs. So this enzyme is not GP, but GH. Next, we performed X-ray crystallography of this enzyme to elucidate the structure-activity relationships. We successfully determined the crystal structure of alpha-1,2 glucosidase. This enzyme was found to form a hexamer. This is a monomer of this enzyme. And as you can see, the whole structure resembles non-bacterial GH65 GPs, cogibiose phosphorylase and maltose phosphorylase. We also analyzed the structure of this enzyme complex with glucose product, glucose in its active site. Active site is the center of this domain. This enzyme has 2 glutamate residues in the active site. They are thought to be essential for hydrolytic reaction. And we identify the residues which are involved substrate binding. So we relieved how this enzyme recognizes and hydrolyzes cogibiose in the atomic level. Next, we would like to introduce the second study, structure function of 31 alpha-1 3-glucosidase. GH31 family comprised of enzymes active on alpha-glucosidic linkage, such as alpha-glucosidase, like intestinal maltase, and xylosidases. This is the phylogenetic tree of GH31 proteins. Similar to the previous study, we targeted the protein that belonged to function unknown protein cluster and have low sequence identity with reported GH31 enzymes. We clone a gene from Lactococcus lactis. This is lactic acid bacteria and expressed in E. coli, and then investigated its activity for glucose disaccharides and sucrose. This enzyme hydrolyzed alpha-1,3 glucobiose nigerose most efficiently, and also hydrolyzed cogibiose and maltose with lower efficiency, although most of known GH31 alpha glucosidase have the highest activity towards alpha-1,4 linkage of maltose. We also determined the crystal structure of this enzyme using X-ray crystallography. This enzyme also forms a hexamer, and its monomer resembles the known GH31 enzymes, including human alpha-glucosidase, human intestinal maltase, and human lysosomal glucosidase. In addition to the crystal structure, we had the opportunity to analyze the sample using cryo-EM. This is a cryo-EM machine. We successfully observed the hexameric structure of this enzyme at 2.73 angstrom resolution. This is a relatively high resolution. And the whole structure was almost identical to the crystal structure. We also analyzed the structure of an inactive mutant, D394A, of this enzyme, complexed with substrate nitro oligosaccharides, also determined by X-ray crystallography. This figure shows the nitrotriose bound structure. Nitrotriose was observed at the active center here. We identified the residues which bind nitro oligosaccharides substrate. Nitro oligosaccharides is green one. This enzyme strictly recognizes nitro oligosaccharide by hydrogen bonds and hydrophobic interaction. Interestingly, tyrosine 99 here, this is located in the neighboring protomer in the hexamer. This residue is involved in substrate recognition. So the hexamerization is important for its enzyme activity, enzyme specificity. Okay, this slide shows the conclusions. By the phylogenetic analysis of the GH families and mining bacterial genomes, 2 novel bacterial alpha-glucosidases were discovered, and we revealed the molecular basis for the substrate specificity and reaction. About GH65 alpha-1,2 glucosidase, this is the first bacterial GH belonged to this family, and we reported the first 3-dimensional structure of GH belonging to GH65 family. Because there had been no report on enzymes specific for cogibiose, a new EC number was assigned to this enzyme. Next, GH31 alpha-1,3 glucosidase. Its specificity for alpha-1,3 glucosidic linkage is higher than any other alpha-glucosidase from prokaryotes and eukaryotes. This is also first 3D structure of bacterial alpha-1,3 glucosidase. Alpha-1,2 glucosides and alpha-1,3 glucosides are rare in nature compared to alpha-1,4 glucosides like maltose and starch, and these components found in polysaccharides produced by some bacteria and fungi. So we think that these enzymes maybe contribute to degradation of such polysaccharides. We will continue to search for GHs with novel activity and elucidate their structure-function relationship. And we aim to apply these enzymes to synthesis and processing of useful carbohydrates. These researches were supported by nice collaborators. Thank you for attention.
Thank you, Dr. Miyazaki, for the presentation. It's very great to see your results during your activity in Shizuoka University. So we have several minutes for the next session. If there is some question from the audience, we have like maybe 3 minutes. If not, maybe I have one question to Dr. Miyazaki. So as you mentioned that GH mainly have some activity to catalyze or hydrolyze the glycosidic bond in complex sugar. And we know that the function of this enzyme in nature, including degradation of biomass such as cellulose or starch. And I found also there is another function of this enzyme regarding in antibacterial defense strategy, or even in pathogenesis of in some viruses. So in your future study, is there any some other approach to see the function of this enzyme regarding for like antiviral activity by using this enzyme? Is there any possibility to use this enzyme for this kind of activity in the future?
I think when the harvest on some polysaccharide
derived from pathogenic bacteria, we can degrade these polysaccharides using specific GHs in the future.
Okay. Yeah. I think that's a very interesting point. We can use this kind of enzyme not only for industry but also for activity. I think also that's possible. I think maybe in the future. So if there is no other question from the audience, I will close this session from Miyazaki. So please close to him. Thank you very much.
To him, thank you. Terima kasih.
Thank you. Arigatou gozaimasu. So next to session, I'll go back to Dr.
It was so lovely presentations from Dr. Miyazaki. Next to the presentation sessions, this time will be led by Dr. Tarwadi, where he himself is the lead of research center of adjuvant therapy. So now floor and time is yours, Dr. Tarwadi. Please go ahead.
Check. Is it okay? Yeah. Okay. So let's start again. So, ladies and gentlemen, we have a second session, and we have approximately 1 hour for 3 speakers. So hopefully every speaker will spend about 15 minutes. And the first speaker is Dr. Engineering Saikhul El-Mutakin. He belongs to the Research Center for Vaccine and Drug. And right now, he's doing postdoctoral in Shizuoka University. He will present polyaniline-based electrochemical impedance spectroscopy, biosensors for virus detection. So, Dr. Rizal, please, the time is used roughly 15 minutes.
Selamat pagi, Assalamualaikum warahmatullahi wabarakatuh. Thank you for everyone. Thank you, Pak Tarwadi, for the introduction. Today I would like to presenting about my research entitled Polyaniline-Based Electrochemical Impedance Spectroscopy, AIS, Biosensor for Virus Detection. My name is Rizal. I'm currently doing postdoctoral period in Park Science Laboratory, Shizuoka University. So this is the very short introduction about as you know, the infectious disease as the main cause of significant increase in pathogenesis and death throughout the world. And we're just suffering from COVID-19 right now. And the golden standard for virus detection is like a viral cultivation, ELISA, and also what we right now know better, polymerase chain reaction, or RT-PCR. But as we know that those golden standard method still suffering from some disadvantage, like a strict laboratory condition requiring well-trained staff and expensive instrument, and also time-consuming process. Therefore, biosensor offers rapid, accurate, dependable, and miniature device for virus detection that probably will improve the capacity test, while in the future we will have another pandemic. We hope not. We hope not, of course. So this is the kind of the simple overview about the electrochemical biosensor. Since the beginning of introduction of the glucose biosensor, actually the biosensor for infectious disease become more famous. And as you can see here, the illustration is almost similar what we already know about the antigen COVID-19 swab test. It's basically kind of the decoration of the electrode here. And we have electrode, of course, because it's electrochemical sensor. We need electrode that conductive that could deliver the electron transport. Here, it probably could be a carbon-based nanomaterial or non-carbon-based nanomaterial. And then after that, we need some kind of the binding molecules. It could be enzyme, antibody, or the single-stranded DNA that could recognize the protein target that we will like to detect. And at the end, after the complex form between the antibody, for example, and also the antigen here from the virus target, we could detect the complex formation by, of course, because it's electrochemical, we could detect it by some parameter like a current, potential difference, and impedance. And here I want to focus about the electrochemical impedance spectroscopy, because among the others method here, electron impedance spectroscopy offer more sensitive method for detecting virus. This is the simple illustration about the AES itself. So basically, it's almost similar like what the others method for detecting the infectious virus. Here, once the complex between the antibody here or the biomolecule recognition might already formed with the virus target, then we could detect the response. Of course, it's quite quantitative response here. Once the higher amount of the protein bind with the antibody, for example, here, the higher like a semi-circular response form in the device, or the AES device. So basically, we could detect and also we could quantify how many virus bind into the device. This is the concept of the AES measurement for virus detection. So here in Park's laboratory, they are quite well known and also expert for fabricating some of the AES-based device for detecting of the virus infectious disease like a norovirus here, and also mosquito-borne virus like chikungunya and also dengue, and also hepatitis virus here in Nature Communication 2-3 years ago, and also even cancer cell. So basically, here I'm coming to the Park Sensor Laboratory the beginning of this year, April 2022, and my task is for fabricating this AES biosensor for virus detection. For fabricating the biosensor for AES and also other electrochemical biosensor, the nanowire or conducting polymer is very important because they will deliver the electron for recognizing the response once the virus bind to the antibody. And here in Park Sensor Laboratory, the polyaniline is very useful, that has been applied for the several biosensor platform. And however, the limitation of the polyaniline here is the emeraldine salt is only conducting state of the polyaniline, which can be obtained in acidic condition, which is almost impossible if we want to construct the antibody-based biosensor in this quite slightly acidic condition. But we could modify the polyaniline and also improve the conductivity of the polyaniline by depositing like gold nanoparticle or quantum dot or another moiety that could improve the conductivity of this polymer. Here, my task during my postdoc is for fabricating the simple stable polyaniline-based electrochemical impedance for norovirus detection. Is quite famous and also infectious diseases in Japan, especially for elderly. And this is the illustration how I construct the electrode for the biosensor norovirus detection. So basically, this is the electrode itself. This is kind of the reusable electrode. We call it gold electrode. This is the gold electrode before we depositing before we modifying with antibody. Here is quite like gold, very shine, and also have a mirror-like character. And then after we have the gold electrode, we depositing with polyaniline here, the green one, and then it's the electropolymerization of the polyaniline, and then we are depositing the kind of nanocomposite between the polyaniline and also gold nanoparticle. The character of the polyaniline, it has a secondary amine here, NH, that could be later on we could deposit or we could modify the antibody here by activating the carboxylic acid group in the antibody by EDC-NHS. Then we are blocking the empty space here to minimize the unspecific interaction. Then we are measuring the AES response by this AES machine here. And yeah, in total, this is the kind of the illustration how long it takes for the detection. Then this the first step. We have to prepare the polyaniline gold nanoparticle nanocomposite. Is kind of the simple mixing between the polyaniline and also the gold nanoparticle. Here I'm using several size of nanoparticle, 520, 40, and 80 nanometer. And this is the temp result here. The black dark color is belongs to the gold nanoparticle, and yeah, the gold nanoparticle size is less than 10 nanometer here that I'm using. Then this is the kind of the typical Nyquist plot that we get when we are detecting the virus. This is usually once electrode will be scanned 2 times. Once before the virus will incubated. See this kind of the small peak here, and then while there is a virus added into the electrode, the curve will be greatly increased. The radius of the semicircle Nyquist plot will be increased. So this is basically the concept of the AES detection. What I want to show here, the next slide here, is how gold nanoparticle improve the conductivity. Here, the higher radius of the circle here, indicating that the impedance or the resistance is very high. While here the two graph here, the upper graph here is only polyaniline without any gold nanoparticle. while here the below small one here, it is the polyaniline and gold nanoparticle nanocomposite, which has much significantly a small radius of the impedance here, indicating that gold nanoparticle is indeed very important for improving the conductivity of the sensor. Then I'm trying to make a kind of a calibration curve, and then checking how if we change the gold nanoparticle size in response to the virus detection. And as you can see here, I'm also checking the nanocomposite without any gold nanoparticle, and there is just stop for a while. Okay. May I continue? Yeah. Okay. So yeah, this is the trend of the calibration curve when we are increasing the concentration of the virus and evaluate the dose-dependent response from the IIS. And this is the regression curve. And then what we could see here is 5 nanometer give much lower limit of detection compared to the others nanocomposite. Here, after getting the good result from the previous slide, I'm checking the platform that I have been fabricated by detecting the norovirus, and I'm immersing the nanovirus sample in the human serum, and this is what we got. Is there is kind of very strong dose-dependent response. If we are increasing the virus concentration, the response also increase. While interestingly, if we change into the dengue virus, there's no interaction, no significant interaction, that's indicating that very high specificity of the sensor. Then I'm also interested to check the platform, the same platform for detecting the dengue virus, not only norovirus. And again, there's kind of the dose-dependent response. While when we are using the platform for detecting norovirus, the very low response was observed, indicating again the specificity of the response. So next, I would like to changing the platform from gold electrode, which is a reusable but quite big platform, to the ones which is disposable but quite handy, and also they're disposable and also portable, using and detect and using this portable ASMS instrument. From the preliminary result, I'm trying to fabricate the electrode for detecting the dengue virus, and it is from our preliminary result. Then yeah, it's quite nice still from the single experiment that could be improved later on. Then this is the feature direction that I want to improve in the future. It's basically in the conventional biosensor, we are still need antibody for detecting the kind of the target virus. But here there is so many research that currently have been performed by constructing molecularly embedded polymer-based biosensor. So it could be some people call it kind of the plastic antibody. So basically, we are using template here. If we want to detect dengue virus, we are using the dengue virus here. We don't need any antibody here, and then the dengue virus will be polymerized with another monomer, and then it will be like a kind of the hollow vicinity that will be exactly fit with the target molecule. And then we could remove this one, and then remove the template that we use for forming this kind of vicinity, and then we could using this platform for detecting the virus. So basically, no need antibody for the detection. Then yeah, for the conclusion, sensitive, stable, and universal biosensor platform is important for dealing with the spread of infectious disease, what we are facing during the last pandemic for COVID-19. And then, of course, the existing collaboration between the current Shizuoka University and BRIN improve our national research capacity to develop biosensor platform for detection in infectious disease. And I just want to say thanks to the Pak Sensei for allowing me to join his laboratory, and also BRIN to allow me to go postdoctoral in Shizuoka University. Thank you very much.
Thank you very much, Dr. Saiful Rizal. Indeed, it is very interesting and stimulating many things in our mind. But since we still have another presenter, so let's move to the second one. So for you who have several questions, please keep in your mind and asking later on. So our second presenter will be Indra Memdi Khois, Master of Agriculture, is actually PhD candidate, hopefully soon will be a doctor. And he will present a nano catalyst and its application for virus detection. So Indra, a PhD candidate, the time is yours.
Yeah, thank you for the friendly introductions from Dr. Tawadi. I'm very is it audible? Is it audible?
Yes.
Okay. So my name is Indra Menukus and I ask permission to share my slides. Okay, is it?
Yes.
Okay. So good morning, everyone, and thank you for the opportunity for me that I can present my works and interest in this mini symposiums together with BRINs. I mean the great institutions in Indonesia, and with all the fellow researchers. Today I would like to present my interest. Today I would like to introduce my research interest in this limited times. So let me use pointer. So my name is Indra Menukus. Again, I have to introduce myself. I was a PhD student, which I will get my PhD titles next month, I hope. I'm from the Department of Bioscience, a graduate school of science and technology from Shizuoka University. I have my master degree in the same university and my bachelor degree from Surya University from Tangerang. And my big title is Nano Catalyst and its application for fire detections. So there's three key points here. First is nano catalyst, then virus and detections. So each of them I will introduce in the overview. First is what is the viruses and as infectious disease agents. I think we all know about this because of the pandemic of SARS-CoV-2. And the second one, I will a bit introduce about immunoassays as a tools for detection of virus. I think it's also very common and very general for everyone because of the commercialization of the antigen kits from BRIN as well. And the third one is about nanocatalysts, which in here specifically is about nanoenzymes. Then nanoenzyme in immunoassay applications. Then how a heterostructured nanomaterials as applied material science is used as an enhanced nanoenzymes and its applications in the immunoassay. Let's proceed. The first one I would like to introduce. So why is virus is the main target in the infectious disease agents, especially in the virus causing disease. So virus, there's two big targets or big factors in here is the antigen or nucleic acids. Of course, there's a lot of biomarker out there, but we will now narrow down to an antigen and nucleic acids detections. And I'm choosing an antigen as the target of interest in the immunoassay because of non-pretreatment required to release or to get the nucleic acid inside the viruses. So antigen is the interesting target for a simple and rapid test detections. Then the final target is the fabrication of on-site biosensor, which should include a rapid, reliable, and cost-effective features inside the biosensor fabrications. So the principle of the detections generally is based on the immunoassay. It means that an assay based on immunology body in here is based on the antibody, we can say, which in the ELISA, an enzyme-linked immunoassay servants, the most common immunoassay that was available in the market and in the stage of development, is using a peroxidase, is an enzyme that was will have an activity that decomposes S2O2 and oxidize a substrate that shows a color. However, as a generator of in the immunoassay, peroxidase, or most commonly horseradish peroxidase, HRP, have a difficulty in the productions and purification. Recently was highlighted, although previously they highlighted on the instability in the harsh environment due to its proteins family. I mean, as an enzyme is of proteins, then it was quite unstable in a harsh environment. Hence, we promote that if we can enhance or if we can change the enzyme into a nanoenzyme. So what is actually nanoenzymes? Actually, it's coming from a nanomaterials that exhibit an enzymatic reactions or enzymatic activity. So from peroxidase, we propose a gonal particles, a nanomaterials with a nanoscale size that have a similar activity that oxidize a substrate that was colorless initially into producing a color as an oxide form, which with the presence of a hydrogen peroxide or a peroxide species, which shows a tunable activity. This is more important one: tunable activity, resistant, and a high surface specific area. So in the immunoassay, the concept is similar as in the common ELISA, in which that we prepare first with fabricating the platforms with the capture antibody and etc. Addition of the virus to make a bonding conjugation between the capture antibody with the viruses, and with the nanomaterial that we introduce, or we call it nanoenzyme here, it will source a color change due to the presence of this sandwich assay. But however, in the story that we want to functionalize the nanoenzyme for an immunoassay, because the nanoenzyme is just a nanomaterials, we need to conjugate it with an antibody for it to be applicable in a specific immunoassay against our targets. Hence, we need to function with antibody, which we proved by ELISA that we can have antibody on the gonal particles, but not in the one that we did introduced. Similar with another ones that we use anti-HB and anti-norovirus. However, we found that in the previous study that conjugating or functionalization of nanoenzymes pacified or passive patients of the surface of these nanoenzymes, causing a decrease of activity in its application as a nanoenzymes. Hence, we introduced two methods in our research group, using a heterostructure nanomaterials as enhanced nanoenzymes. So what is heterostructure? It means that a nanomaterials which two kind of components, which we call heterostructure or heterocomposite. In the first one, is published in the BNB 2019, you combining gold nanoparticles and silver nanoparticles as the coating on the surface of the gold. After the conjugations, we call it silver shell deposition on gold core, which shows a high catalytic activity. And the second one, I will introduce also our recent works in a heterostructure using a platinum nanoparticles and the nano cage using MOF, metal oxides nano cage. So the first one, short introduction of the application in an enzyme using the heterostructure using gold and silver. We can see that in here is gold initially, then we can coat it the silver on the surface of the gold, showing a double different structure in the TM, and using the absorbance as a visible light, we can see different absorbance clearly showing the coating on our surface, and we successfully detect norovirus and hepatitis E virus as a monitoring functions based on the RT-PCR functions on the monkey. And we also compare the black dots here, compare with a commercial ELISA, or actually a conventional ELISA, showing higher linear calibrations on our sites using our enhanced nanoenzymes immunoassay. Proceed. We also would like to introduce the second heterostructure. Similarly, with after the immuno sandwich formation with our heterostructure nanoenzymes, it will exhibit a color change when we introduce our substrates, showing the detection of the virus based on the color. Beautifully, on the TM analysis, we can see the oxidize of the nanocage, showing a porous, semi-porous, or we call it pseudo-porous of the nanocage with integrated platinum within the structure. The XRD shows some platinums difference. Of course, more beautifully, we can see the elemental analysis. The blue side here is platinums within the structure of our nanocages. And here also we can see that the important thing is the B part. We introduce a not novels, a derivative factor to examine the nanoenzyme in the immunoassay based on the derivative of the kinetic parameter of non-enzymatic reactions, together with some sigmoids analysis, sigmoids curve analysis, which is usually used in the drugs, which is we call it IC50. In here we call it NZ50, nanoenzymes half reactions. Then we can indicate that the platinum nanoparticles within the nanocage is much stronger, or I mean, this requires small amount to exhibit a high activity compared to the normal nanocage without nanoenzymes, showing the heterostructure actually improving, showing the concept of nanocage and nanoenzyme together. Further, and lastly, the most important thing, we can detect norovirus. We can directly go to the C, that's in the clinical norovirus that isolated from the human, we can extremely compare our results to the commercial kit from the local company in the Japan, that it can show higher linear, wider linear calibrations with higher sensitivity compared to the ELISA kit on the same concept of immunoassays, but using different enzyme and nanoenzymes. Selectivity on the B side, showing only norovirus detections, is only because of the presence of norovirus, it will change color, not in other proteins or viruses. And the D1, we can both detect SARS-CoV-2 in case of spike protein and N proteins using specific antibody. So by changing the antibody, we can actually detect specific virus using our nanoenzymes. So in the conclusions, I hope I didn't speak too fast, or the conclusion is that we want to detect initially plasmonic chromatic is used as the what's double sum? Used in the commercial kit. Then it go with the super enhanced, published or commercialized by Fujifilm company in Japan. Then we think that it can be go further as nanoenzymes for an integration available platform of immunoassay with nanoenzyme concepts. This is our nanobio groups in the Park Labs University. I thank you for the research fellowship from GSPS and the collaborator that provides the viruses and VLP for our detections. Other works in our nanobio groups, some gold positive charge nanoparticle and nanoenzymes, supported gold nanoparticle nanoenzymes in a 2D structure, some encapsulation concept with the gold nanoparticles and some metal oxide nanoenzymes. The last one is electro nanocatalyst for fire detection. That's all. Thank you for your attention.
Thank you, Dr. Indra, Dr. Candidate Indra. So, ladies and gentlemen, actually, in the presentation is very promising and potential to be fabricated. So hopefully in the next future, we will have another product which is very sensitive to detect virus infection. Ladies and gentlemen, now we came up with the third presenter. Here's Dr. Dodi Irawan Satyo Utama, belong to BRIN organization, and he's graduated from Shizuoka University, and he will present his talk regarding humoral immune response induced with monovalent and tetravalent dengue virus-like particles produced in silkworm. So, Dr. Dodi, please, time is yours.
Thank you.
protein itself for the envelope particle.
Okay. Sorry, I will. Should I restart? Continue. Continue. Okay. Sorry, I forgot to unmute. So really sorry about this. So I will continue about the virus particle itself. So it's a self-assembly and also like in the viral genome. Because, that's why it's become non-infectious compared to the real virus and have a high stability because it's like the real virus itself and can be composed, can be polyvalent structure and have a size in the range of 20 until 100 nanometers. And this can be easily to modify and have a homogeneous in the display of the epitope itself. So in here to produce the virus-like particle, I use the BMNPV or Bombyx mononuclear polyhedrovirus for the silkworm expression system. In here, why I use the silkworm expression system, because this is stable in the production of the protein, and it has a post-transitional modification. Silkworm itself can produce a high level of the secreted protein, and it's easier to maintain, doesn't need aseptic condition, and because this is a very sustainable protein expression system, and it's also environmental friendly. That's why I choose this Bombyx mori to produce the PLP. So for my research outline for the monovalent and the tetravalent, so I start from the monovalent, which is I construct the cpRNA and pRNA. And this is the I use the synthetic DNA, which is this is contain the C capsid, premembran, and envelope, and also the premembran and envelope of the dengue, and express this to, using the silkworm expression system and purified using the affinity chromatography, and did the biological functional assay for the dengue LPs that form using this construct. And then after a biological functional assay, I continue to do animal experiment to see this BLP can induce the humoral response in the mice. So while for the tetravalent, almost similar the flow of the research, but in the to do the expression, I did the co-expression for the construct cpRNA and pRNA, and this is I will explain later how to do the co-expression. Meanwhile, for the other flow is almost similar until for the humoral response for the this dengue VLP in the mice. So I will continue with the monovalent first. So in here, I construct the genes of each serotype of the dengue, which is contain, I already mentioned, it's contain the capsid, premembran, and envelope. And in here, at first I produce the plasmid recombinant, each of the serotype, and then produce the backmid recombinant. And from with the backmid, with this backmid, each of the backmid is injected to the silkworm, and the silkworm will produce the monovalent dengue VLPs. So this is for the expression itself. In the red arrow, it's shown that the protein is expressed. First, I tried to express it in the larva, and it's expressed in the larva and can secrete it to the hemolymph. And the other is I also did it in the pupa, and it also can be expressed in the pupa for both of the construct, and also in the BF5 cells, which is this is the other type of the expression system included in the silkworm expression system. BF5 cells is the cell line of the Bombyx mori itself, and it can also express my protein. So both of the construct for the CPRMA and PRMA can be expressed in the Silcom expression system. For those one is for the dengue 2, while for the other dengue 1, 3, and 4, I only focus in the expression in the larva, which is in this red arrow. It's shown that the protein is expressed and secreted to the hemolymph. And for additional, I also express the E subunit for all serotype. This is for the ITC. This is for the heparin binding assay. So yes, from this we can see that all of the protein can be expressed. So after that, using the affinity chromatography, flag tag, I tried to purify the both of the construct of the protein. And in here, we can see the band in the red arrow that all of the serotype can be purified using the using the affinity chromatography, including the subunit. So it means that these all of the protein CPRMA and PRMAs monovalent can be purified by the affinity chromatography. While the dengue 3, because the dengue 3 for the tag is kind of different, I use the PA tag. It also can be purified using the PA tag affinity chromatography. And to see if the construct that I use, it can produce or form the PLP, we need to do the transmission electron microscopy to see the form of the PLP itself, and to check if this PLP can display the epitope that we want. So composed by the epitope or the envelope of the dengue itself. So from here, I use the first antibody, which is this is in here. I use the polyclonal antibody of the dengue, envelope of the dengue, and the second antibody for the HRP. And in here, the HRP is conjugate with the gold nanoparticle. So this can show the surface of the BLP is contain the envelope of dengue or not. So from the result for each the serotype in here, for each for each the construct, we can see there is a small dot, black dot on this structure. So this structure. So this is the gold, conjugated gold nanoparticle, which is this bind to the first antibody. The first antibody bind to the envelope on the surface of the dengue, and this is we can see that this my PLP can form, my construct can form the PLP, and this PLP have the envelope on the outer side of the PLP itself. So to continue, if this PLP contain the ED3, because the ED3 or envelope domain 3 is the infectious epitope region in the dengue. So we need to do the heparin binding assay for this, because the heparin can bind to the ED3 site of the dengue. So from here, I use the afidin coated microplates and bind with the biotin heparin, and put my antigen and detect with my first antibody, which is the anti-E, and also the SRP conjugated antibody. And here we can see from the concentration of the protein, and it can bind with the heparin until 20 microgram per milliliter. This is also this is also content for all the serotype. So from the heparin binding assay, we can see that the VLP is contain the ED3. To make sure this ED3 can bind perfectly to the heparin receptor in the in the cell, we need to do the ITC to see the affinity binding between the heparin receptor with the ED3. And in here, we can see there is a sigmoidal graphic, which is this is indicate that the binding is can be count, and it's in the good term of binding. So we can calculate and the KD value for this, and for the KD value for the monovalent, I can get in the range of the nanomolar for this, and this range is in the range of the good binding affinity between the heparin and also the ED3. So in here, from the CPRME and PRME binding between the heparin and also the ED3. So we can see the ED3 is present on the surface and the monovalent of the PLP. And for the value, the KD value for the CPRME is lower than the PRME, which is CPRME have a higher affinity to the heparin compared to the PRME. And we compare also for the subunit only for the E, which is this is produced by the silkworm. We compare with the E from the dengue to produced by the cell of COS by COS7, and the value is can be comparable from the protein of envelope dengue 2 to the envelope dengue 2, which is produced in the mammalian cell. And to see if this VLP have a real have a similar epitope display with the natural dengue virus, we challenge this. We we challenge this with the mixed sera dengue patient or human human patient. So in here we can see the binding between the human between the patient sera with the VLP is higher compared with the with the control of anti envelope. And in here we can see that the this VLP have a similar epitope with the natural dengue virus. To see if this VLP can induce the humoral response in the mice, so we inject this. We do the animal experiments for this in the mice. So we inject the this VLP in the biopsy and immunize it for 28 days with the 2 booster. And from the blood that we isolate, we can see that in here the graphic in the graphic that the antisera from the mice it can be bind with their own antigen. So and in here we can see that the CPRME have a better response compared with the compared to the PRME. And for others is we also use the adjuvant to see if this adjuvant can help the response better compared with without the adjuvant. And we can see in this that the with the adjuvant have a better response compared with without the adjuvant. So for the tetravalent vaccine candidate, from the structural gene that I already construct for the monovalent, same I use to produce to make the plasmid recombinant and produce the backbone recombinant. But in here I mix all the backbone recombinant by ratio 1:1:1 each. So in here these 4 directly injected to the silkworm itself, and then we assume that this will produce the tetravalent dengue VLP with the variation of the backnit. For first, we need to check the expression itself, and in here, by detecting with the anti-E polyclonal antibody, we can see there is a band, expression band in the hemolymph, and we also need try to check if this construct, the CPRME and PRME co-expression, have all of the epitope or the variation of the envelope from the each serotype. So in here we can see that each serotype, its construct is detected by the antibody from each serotype, and we can see there is a expression in here. We can see there is a band in here. So this is we assume that this expression can be confirmed and have a variation of the envelope. And we need to purify this expressed protein. And in here, I use the magnetic flag tag purification, which will save time and also have a better amount of the purified protein compared to the conventional affinity chromatography. So from here, we can see there is a band indicated by the red arrow, which is detected by the polyclonal antibody from the purified protein from its construct. Again, to confirm the variation of the envelope itself for all serotype, I detected using this different serotype of monoclonal antibody, and it can be confirmed. So this means that its monoclonal antibody can confirm the display of its envelope in this both of the construct of this tetravalent VLP. Again, to see this VLP this construct can form the VLP, I use the immunotherm with the same way with the monovalent. But in here, I'm not only detect using the polyclonal antibody, but also I confirm with the monoclonal antibody. And in here, we can see there is a little black dot in the attaching to this circular spherical structure for both of the constructs, CPRMA and PRMA. And this also confirmed by its serotype monoclonal antibody, which is its monoclonal antibody. In here, we can see the bind of the gold nanoparticle to the surface of the VLP. And in here, this construct capable to generating the VLP and also can display for the variation of the serotype. Same to see the ED3 on the surface of the VLP itself. I need to do the heparin binding assay, and from here we can see same pattern, which is the binding of the heparin to the surface of the VLP, and also similar to the ITC result itself. And from the ITC result, we can see that the value if we compare with the monovalent, it's lower than the monovalent, which is this also confirm that the ED3 is present on the surface of the tetravalent VLPs, and the CPRMA tetravalent have a better higher affinity compared with the PRME. And same, we also try to detect this, to bind this with the Mixera dengue patient. And in here we can see compared to the positive control, that the patient have a higher response compared to the only with the anti-envelope polyclonal antibody. So means that this VLP, tetravalent VLP, have a similar epitope to the natural dengue virus. So again, to check the humoral response from this VLP, we need to inject to the mice and produce the antibody, and from the antibody produced by mice, we detect with the antigen, and this from here we can see the CPRMA compared with the PRMA have a higher response, and with the adjuvant also can increase the response for the humoral in the mice. So for the conclusion, for all the monovalent and tetravalent CPRMA and PRMA, it can be formed, and this VLP have a proper structure with the ED3 present on its surface, and this VLP shared a similar epitope with the natural dengue virus, and the tetravalent dengue VLP elicits more immune response than the monovalent. And this, I would like to send my special gratitude to my professor, Professor Eno Park, and also Dr. Sabar for their guidance during my study, and also Dr. Fitya Sata from University of Indonesia, and Dr. Etik Mardiati, Mr. Ali, Nur Ali, Mr. Joham, and also the all member of the Laboratory of Biotechnology in Suzuka University, and also the members of the diagnostic kit lab in BRIN. Okay, I think that's all for my presentation. Thank you very much for your attention.
Thank you so much, Dr. Dodi. So many things, so many data have been presented. So now we end up with the question and answer. I think we're running out of time, but if you have any question, then please raise hand and directly to the speakers. Okay. Yes, please say from Mr. Ivan Mambaunir. If you would like to open mic and speak directly, maybe better. I just already know. Okay, please. Okay, please. Miss Karidat Bihandayani. Miss Farida.
Oh, yeah. Thank you very much for the opportunity. I'm sorry. Okay, so I have a question to Dr. Dodi. So for the characterization, what kind of methodology you explain? You explain to characterize your monoclonal. Thank you very much. I'm sorry, you are still mute.
Sorry, it's happened again. So thank you for the question, Mrs. Farida. So for the monovalent and also tetravalent, actually I did the same characterization, only with the different antibody for the because I need to check the the display of the envelope for the each dengue in the tetravalent, but basically same. So for the characterization itself, beside the SDS page and Western blot, so I did also the TEM and also immunoTEM to see the forming of the VLP and also the composition of the VLP itself, which is in the outer side, it will be display the envelope. So to detect that, we need to detect it using the immunoTEM, which is almost similar with the Western blot and also the ELISA, but different is we can see the binding between the gold nanoparticle with the surface of the protein if the envelope is displayed. It also can be done in the tetravalent also. And the other thing is I did the heparin binding assay, which is can confirm the ED3, the envelope domain 3 on the surface of the dengue cell, because this ED3 will bind to the surface of the cells. And the other thing is for the humoral, so I did the animal experiment in mice to see the humoral response for both of the construct and also both of the kind of the VLP.
Yeah, thank you very much. Good luck for your study.
Okay, thank you very much.
Okay, thank you, Ms. Faida. Dr. Dodi, this is a question in here. It's from Ivan Mamba Ulmune. For Dr. Dodi, I'm just curious, why did you use silkworm as your organism model to conduct your research? Silkworm is an unusual organism model, as I know, for gene expression experiment. So please respond it, Dr. Dodi.
Thank you very much, Mr. Ivan, for the question. Why I use the silkworm itself, because the silkworm can have a ability to produce the protein similar to the mammalian itself, and it's easier to maintain because you don't need any aseptic condition, and to take care of the silkworm, it's quite fast to produce the protein itself. And you don't need many amount of the, like if you produce using the mammalian cells, you need much amount for the cell for the expression, while silkworm you only need several silkworm to have a same concentration of protein that you can get compared with the mammalian cell. And the other thing is the silkworm is sustainable, so and it's safe for the environment. I think that's what I can answer.
Okay, thank you, Dr. Dodi, for the response. So I think if there is some other question, please raise hand or just write it down in the box. Okay, so this is because we are running out of time. So just another question. I'm very sorry for this. Siapa nih? Okay, Bu Winnie. Yeah. This is from Bu Winnie. I would like to ask Dr. Dodi, how about antibody neutralization? Did you also examine it? Yeah, please, Dr. Dodi.
Okay, thank you for the question, Dr. Winnie. Actually, yes, this is for the neutralization assay. It's ongoing. But some of data I already got, and it's also already presented in my defense. But I cannot put in here because all of the experiment is not yet finished. So I would like to finish this as fast as I can for the neutralization assay during my time in BRIN here now. So yes, it's in progress. Thank you.
Thank you, Dr. Dadi. I think there is some still question, especially for Dr. Rizal and also for Mr. Inda. But since we are running out of time, so I'm very sorry to close this session. So please give a big applause for our presenters.
Thank you so much.
Thank you so much, and I would like to turn it to Dr. Eka. Thank you.
Seems like we all heating up already in this session, on the second session. So the highlight is actually we go from the basic to the applications, and we have gone through all most recent update in regard with what's it? Biosensors and diagnostic tools. We are going to move on very much downstream application of another application that is more into antibody monoclonal, which will be facilitated by Dr. Astuti Nurhasana. Pardon me. So, floor is yours.
Okay, thank you very much, Dr. Ika Nurlaila. Good afternoon, ladies and gentlemen. It's an honor for me to lead this session, who is an all-ladies session. Since we are actually almost half an hour behind the schedule, I would just like to introduce the three speakers that are going to deliver their speech today. The first one is Ms. Tika Widayanti, from Center for Vaccine and Drug Research, and then the second will be Ms. Asri Sulfianti, and the third will be Ms. Feby Nurdianingsih, also from the Center for Vaccine and Drug Research. The first one, Ms. Tika Widayanti, has got a master's degree from the Faculty of Medicine, University of Indonesia, and at the moment she's going to deliver her presentation in the development of monoclonal antibody anti-spike SARS-CoV-2. Please, Ms. Tika Widayanti.
Thank you, Buti, for your kind introduction. Good morning, ladies and gentlemen. My name is Tika Widayanti. I would like to present our recent research about development of monoclonal antibody anti-spike SARS-CoV-2. SARS-CoV-2 virus is called coronavirus disease that spread globally and became the major health problem since today, with the death tolls worldwide more than 6 million people. On the left, it's the genome of SARS-CoV-2, which contains 4 structural proteins, including spike protein. The spike protein plays the key role in initiating viral integration into the host cells. These important roles made the spike protein as a target for drugs and detection. So what is a monoclonal antibodies and why we should develop this molecule? Basically, monoclonal antibodies or MABs is a monospecific antibody that selectively only bind to one epitope. The advantages of MABs are MABs can be produced in laboratory, can be engineered, modified, and its properties mimicking natural immune response. MABs has been used widely as reagents in research field for diagnostics materials such as lateral flow assay or immunodiagnostics, and therapeutic applications for cancers, infectious disease, and so on. The picture on the right is one of the methods to generate MIBs. The fusion between B cells from mice, splenocytes, and tumor myeloma cells resulted in hybrid cells that can produce MABs. Therefore, in this study, we generate a monoclonal antibody for spike protein from SARS-CoV-2 that have crucial role in penetrating host cells and initiating infection. In order to generate hybridoma MABs producing cells, the first is to clone and express the spike RBD protein in E. coli. Hereafter, the recombinant protein was injected to biopsy mice with a 2 weeks interval. Hybridomas were obtained by the fusion between myelomas and B cells from immunized mice. After screening for potential hybridomas, MIBs were produced by injecting hybridoma cells back to the mice, or another method is hybridoma cultivation in cell culture medium. The MABs purification is performed by using affinity chromatography. For protein analysis, MABs were tested by indirect ELISA and Western blot imaging. So here's the result. Hybridoma cells generated by cell fusion between spleen cell from immunized mice and myeloma cells, as shown in left figure. The screening of potential MABs were performed with indirect ELISA. 5 wells with the highest absorbences were selected for further step to obtain single hybridoma clone by limiting dilution. Monoclonal antibodies were tested against recombinant spike protein and whole inactivated virus. In this study, we use CoronaVac or Sinovac vaccine as antigen source. The hybridoma selection with indirect ELISA showed 17 clones that potentially produce anti-spike antibodies. However, 4 out of 17 MAB clones were consistently shown positive result by ELISA analysis against inactivated COVID-19 vaccine and recombinant spike protein. In order to get purified MABs, purification process is one of crucial steps in MAB production. Purification is aimed to eliminate lipid particles, cell debris, host cell proteins, and other contaminants that can affect